So let me finally talk about the results. So we have, of course, sequenced all these 8 sets from HV3 to HV10. I'm going to talk about the HV10, which is the most recent one, just to give an idea about the numbers. So after demultiplex, remember we're using a DNA barcoding to multiplex a set of pools on the same lane of the flow cell. So after the multiplexing, we got about 13 million reads per pool with an average length of 92 bases. And then we had to remove E. coli and trimming, adapters, and so on and so forth. So we lost a reasonable, well, not terrible amount of reads. So we're down to 9.7 reads, 9.7 million reads per pool, with an average length of 90 bases. Now, a quick back to the envelope calculation would tell us that given that the average back length in that particular set is about 128 kilobases, this will give a sequencing depth before the convolutions about 500x, which is sufficient to give good assembly of these BACs. Before we talk about the deconvolution, I want to comment on something that we recently discovered. So we look at, we were struggling with some of these data sets, for example, HV9 and other ones. And we were not sure why we were able to decode or deconvolute only a small fraction of these reads. And we suspected that sequencing error were playing a big role in the process of deconvoluting. Sort of someone in the group suggested maybe we should try to look at a fraction of the data and see how we do on this fraction of the data. And in this plot, what we are plotting is we are plotting the percentage of reads in those pools that are actually decoded to a BAC with confidence. And as you can see, for different sets, here we are looking at HV3, HV4, HV9, and HV10. The shape of these curves are different, but they all have some sort of a sweet spot, which is sort of a maximum that is reached between 1 and maybe 2 and 2.5 million reads per pool. So this suggests that actually trying to deconvolute or decode all the data at once is actually not a good idea. So again, the x-axis is the number of reads per pool on average, so in millions. So 1 means 1 million reads per pool, 2 means 2 million reads per pool. So as I said, there is different sets at different sweet spots. And for example, HV9 max out in terms of the number of decoded reads about 1 million reads. So what we did is cut the data set into pieces and then decode each piece individually and then try to combine the solutions, the partial solution, to obtain the final decoding. So the bottom line is that if you have too few reads, of course, you cannot deconvolute because you don't have enough information. But if you have too many reads per pool, then because you have sequencing errors, then it will affect negatively the decoding or the deconvolution, and you don't want that either. This result is still not published. We are writing up a manuscript probably very soon. So given this approach on HV10, we were able to decode about 85% of the reads, which ended up translating into about, again, about 500x coverage. Just to give you an idea about the resources, the computational resources needed to do this job, we're talking about 7 hours on a sort of average server, which has a significant amount of RAM on it. So we used about 36 gigabytes of RAM to do the deconvolution in this case. So it's something that you cannot do on a regular PC, but you need access to a decent server with a multi-core server with a significant amount of RAM. In terms of accuracy, for HV10, we had some of the BAC signature actually were not used. So we had them in the design, but they were actually not used. So there were no BACs actually that employed those signatures. In fact, about 21% of them. And that our tool, of course, we didn't know that, but still did not essentially assign any read to it, or very, very few of them. Only 0.043% of the reads were assigned to these unused BAC signatures, which let us, that sort of suggest that the hash filter process, the hash filter deconvolution or decoding is actually quite accurate, meaning it does not assign a read to the wrong back, which is, of course, would be devastating for the whole process. So we are very confident that the convolution is accurate, and it's a relatively efficient, a few hours of on the server with about 36 K per RAM. So once the reads are assigned to a back, then we can finally assemble them. And again, the advantage of this approach is that we can assemble them back by back. So as Tim mentioned, if you have a sequence which is highly repetitive, maybe it will not be repetitive inside a single back, and therefore we might be able to actually assemble it. So far we've been using Velvet to assemble these backs. And we look at, of course, Velvet has this parameter, which is the hash length. So we tried different choices of the hash length and kept the assembly that has the largest M50. This does not necessarily guarantee that this will be the best overall assembly for those choices of the hash length. But right now, it's sort of an arbitrary choice that we made. For those of you that are not familiar with the term M50, this is the minimum length of all the contigs that together accounts for at least 50% of the target, which is in this case the back that we're trying to assemble. So how does the assembly looks like for HV10? Well, the M50 is very high. The average M50 over the 1,000 back assemblies is a very high. It's about 42,000 base pairs. The largest contigs, again, average largest contigs over all the 1,000 assemblies in HV10 is a 54,000 base pairs, and the sum of all the contig sizes is about 147,000 base pairs, which is sort of overshooting the average back length. So we have some inflation of contigs that needs to be resolved. We also looked at how many bags contain genes that were expected to be contained. So we knew in advance, using a different technology, that some of these bags were expected to contain known genes. And so we looked for those genes in those assemblies. And it turns out that almost 90% of those assemblies contain expected genes. So we did this by blasting, essentially, assemblies against these unigenes. And the coverage is very high. It's over 90%. Just to give you one data point, this is just one back assembly that was sequenced twice, actually, in two sets, in HV3 and HV9. And also was sequenced once as an individual back. So just to give you an idea about how these assemblies compare, or this back assembly using combinatorial pooling compared to if you were doing a single back with just barcoding, right? So we did that. We had one back, this particular back, in fact, more than this back, but this back has been sequenced twice in using combinatorial pooling, once as an individual back. And also, was sequenced using Sanger by Joint Genome Institute. So we can take the Sanger sequencing as what's the ground truth. It was a single contig, so we expected that to be the true answer. And so how these three other assemblies compare to that particular for that particular back. And again, this is one data point, so we cannot necessarily make the same conclusions for all of them, but we think that might be the case. So this table summarizes the comparison. So for HV3, about 600x coverage. So that back in HV9 at 300x of coverage, and as a single back at about 9,000x coverage. When you assemble them using Velvet, as you can see, you have the number of contigs, which actually is the smallest number of contigs in HV9. This back was about 131,000 base pairs. So actually, the assembly in HV9 is the closest to the true size. In terms of the largest contig, the largest contig was in the case of the single, but as you see, the sum is very high. So in some sense, there is a lot of redundancy in this assembly, and we think the reason it might be because the depth of coverage is so high. So we are investigating whether cutting down this coverage, removing some of the data, might actually help with the assembly. The M50, in this case, the highest is for the single. But when we look at misassembly, when we compare these assemblies to the ground truth with the one obtained within Sanger, actually the assembly for HV9 looks the best, as only a 0 misassembly compared to 17 misassemblies of the single back. Also, in terms of the fraction of the ground truth covered by this assembly, actually the combinatorial pooling assembly looks much better than for the single one. And again, these are preliminary results, and I would like, we're still investigating them. We use Quast to do these comparisons, and we hope that these will give us very good indication whether these approaches actually competitive with single back sequencing. Before we finish, I want to mention that these back assemblies are available. We have a website which is linked from the webinar webpage, which is called arabes-web.org. And if you click on the utilities link for barley, you will be able to type in SNPs names in this window and obtain as the output the sequences and the functional notations for any set of BACs in the neighborhood of that list of SNPs. So this is actually goes back to the original motivation of this work that Tim mentioned at the very beginning. We have the very latest version of the BAC assemblies actually are not available at this point, but you can contact us and we can give you access to it. So let me conclude this presentation by saying that this approach of sort of tackling assembly and sequencing of large genomes when they're very repetitive, like barley, might be necessary if you want to obtain high quality sequencing. So we think the back by back might be at this point a very good approach to this problem. But when back by back is sort of combined with next generation sequencing, then you have this problem of handling hundreds or thousands of samples. And because the NGS sequencing machines have a limited number of samples that they can handle, then people use barcoding, but barcoding doesn't really scale readily. And so we think that pooling, this particular type of pooling that we use, is a cost-effective and practical alternative to exhaustive linear barcoding. And in fact, as we mentioned and we used in our work, you can combine the two. You can get the benefits of both, right? And you get a significant reduction in the number of samples that need to be handled. In terms of from a informatics point of view, computational point of view, we're pretty confident that the convolution process, the decoding process, is very accurate at this point. Something that we did not know 3-4 years ago, and that the back assembly is actually a very high quality. In terms of cost, if the NTP set is given, the library NTP is given, the cost is about $10 to $25 a back, which includes pooling, the preps, the sequencing, and the informatics. And as I mentioned in my last slide, the barley bags and the software are available. They're linked from the webinar webpage. This work has been described in a paper in PLOS Computational Biology that came out a few months ago, and we have an improved decoding algorithm that is going to be presented at the workshops on algorithms in bioinformatics in about a month in France. So with this, I want to acknowledge all the people that contributed to this work. Tim, which is here sitting nearby, and in his group, Steve, Rosanna, Joaquin, and Josh took care of the wet lab and the bags and the library and the sequencing. In computer science, myself and Gianfranco Ciardo, we sort of supervise the computational aspect involving the deconvolution, the assembly, and our students, Denise, Matthew, Ray, Yongu, and Serdar, took care of various aspects of these of the computational analysis of the data. We also had help from Francesca and Marco at University of Torino to develop hash filter. In fact, they were the people that eventually wrote a tool that we're still using heavily today. At UC Davis, Chen Lu took care of the fingerprinting, high information content fingerprinting. And as we mentioned, several BACs were actually sequenced using Sanger by John Jenin Institute, and Jeremy Schmutz and Jen Greenwood were the people involved in this work. The support comes from USDA, NIFA, and NSF DBI. And with this, I want to thank all of you for your attention, and we'll be happy to take questions.